Right now, in like roughly a third of the people on Earth, there are these microscopic cysts just quietly sitting inside brain and muscle tissue.
Yeah, completely dormant.
Right, mostly undetectable and just waiting for an event that is, well, probably never going to happen. I mean, they're waiting for their human host to be eaten by a cat.
It really does sound like the setup for some weird science fiction thriller, doesn't it?
Totally.
But for, a massive portion of the global population, it's just everyday biology. We're looking at an organism that is arguably one of the most successful chronic human parasites on the planet.
Welcome to today's deep dive. We have this really fascinating stack of sources to go through with you today, covering the microbiology, the wild history, and of course the intense controversies around a single-celled organism called Toxoplasma gondii.
Right.
And if you've heard of this at all, you probably know it by its internet nickname, right? Like the cat parasite.
Exactly. Or, you might just know it as the reason doctors tell pregnant people to stay away from litter boxes.
Yes, the infamous litter box rule. But our goal today is to cut through all that viral hype.
Because the hype kind of flattens the actual biology. When you really look at the research, Toxoplasma, it isn't just a pathogen causing a disease.
Right.
It's actually a masterclass in evolutionary adaptation. It has a life cycle so complex that human beings barely even factor into its overall strategy.
Wow.
Yeah, we're basically just an accidental, mostly useless stepping stone in its biological journey.
Which is incredibly humbling, right? To realize that to this microscopic organism, we aren't even the main character. We're just a biological waiting room.
That's a great way to put it.
But before we get to how it actually uses us, the origin story of the quote-unquote cat parasite is super fascinating because, well, it doesn't actually start with cats at all.
No, it really doesn't.
The initial discovery back in 1908 is this wild historical coincidence. You have two different research teams on two different continents studying completely different animals, and they found the exact same organism at basically the exact same time.
Yeah, that parallel discovery gives the history such a rich texture. So, over in Tunis, North Africa, you had the French bacteriologist Charles Nicolle.
Who later won a Nobel Prize. Right?
Yes, exactly, for his work on typhus. So he and his colleague Louis Menceau were at the Pasteur Institute, and they were examining this small North African rodent called the gondii.
And that's spelled G-U-N-D-I, right?
Right.
Which gives us the gondii part of Toxoplasma gondii.
Exactly. So they found these strange, microscopic, kind of crescent-shaped parasites multiplying in the rodent's tissues. And meanwhile...
We're literally across the Atlantic.
Literally across the Atlantic, in São Paulo, Brazil, an Italian physician named Alfonso Splendor was investigating a mysterious die-off in his laboratory rabbits.
Oh, wow.
Yeah, and he looked at their tissues under a microscope and saw the exact same crescent-shaped organisms. Nicolle and Menceau just happened to publish first, so their name stuck.
And the name Toxoplasma, I mean, it sounds incredibly sci-fi, but it's actually just a very literal description of what they were seeing through those early microscopes.
Right, because toxin is Greek for bow or arc.
And plasma means form. So arc-shaped form, nodding to that crescent moon shape. But what really jumped out at me in the sources is its family tree.
Yeah.
It belongs to this phylum called Apicomplexa.
Yes.
Which makes it a close cousin to Plasmodium, the parasite that causes malaria.
And that family connection is crucial for understanding how it actually operates. All epicomplexans share this specialized set of organelles at the tip of their cell. It's called the apical complex.
Okay.
You can think of it like a biological battering ram mixed with a lockpick. Instead of just kind of floating around in the bloodstream, Toxoplasma uses this apical complex to actively latch onto a host cell.
And break in.
Yeah, it secretes enzymes that alter the host's membrane and literally pries its way inside. And once it's in there, it's hidden from the antibodies circulating in the blood.
That is so wild. So for decades after 1908, this little arc-shaped lockpicker was basically viewed as an academic curiosity, right? Like a parasite that bothered rodents and rabbits.
Yeah.
But the narrative completely shifted in 1948.
Right, with the dye test.
Yes. Albert Sabin, the guy who developed the oral polio vaccine, and Harry Feldman invented the Sabin-Feldman dye test. This was the first reliable way to check human blood for antibodies to Toxoplasma. And the results from this test completely upended how the medical community viewed the whole thing.
Because the dye test revealed that huge swaths of the human population had actually been infected.
Right.
Before that, toxoplasmosis was thought of as a very rare, usually fatal disease in newborns. But the antibody data proved that millions of healthy adults were carrying it, meaning the vast majority of infections were completely silent.
Okay, let's unpack this. That presents such a bizarre epidemiological puzzle. If you have an organism infecting millions of people, but almost no one is showing symptoms, how do you trace the source? I mean, it's like trying to find the origin of an invisible flood.
It was incredibly difficult. It took decades of detective work to finally map the ecology. The dye test was in 1948, but the real breakthrough didn't happen until the mid-60s and early 70s.
Wow, that long.
Yeah. In 1965, a Scottish parasitologist named William Hutchison demonstrated that the infection could somehow be transmitted through cat feces.
Ah, there's the cat connection.
Exactly. And then in 1970, a team at Kansas State University, J.P. Dubey, N.L. Miller, and J.K. Frankel, they proved the fundamental mechanism.
What did they find?
They discovered that cats, and specifically members of the feline family, are the only animals on Earth that harbor the sexual reproductive stage of this parasite in their intestines.
Because it can only sexually reproduce inside a feline gut, cats are the definitive host. Every other warm-blooded animal, humans, sheep, birds, mice, we are all just intermediate hosts.
Basically, yeah.
We are just biological taxicabs. Like, infecting a human does absolutely nothing to further the parasite's life cycle unless that human is somehow consumed by a feline. Which is why understanding its 3 distinct life stages is so critical, right?
Yep.
Because it has to adapt to survive the taxi ride.
Exactly. Those 3 forms are the absolute key to understanding both the clinical disease and its evolutionary success. Let's start with the form produced in the cat, the oocysts.
I would hate oocysts.
After sexual reproduction in the feline gut, the cat sheds these oocysts in its feces. They are essentially microscopic, heavily armored eggs. The cyst wall is composed of highly cross-linked proteins that make it nearly impervious to environmental degradation.
So they're tough.
Incredibly tough. They can sit in freezing soil or, warm water for over a year without losing their ability to infect a new host.
Which is terrifying. And then once an intermediate host, so let's say a human gardening or maybe a pig eating scraps, accidentally swallows one of those armored oocysts, the stomach acid actually helps break down that outer wall.
Right.
And the parasite hatches and morphs into the second form, the tachyzoites.
Yes. Tachy meaning fast. These are the aggressive, rapidly dividing crescent shapes.
The lockpickers.
Exactly. They use that apical complex to punch into our cells, hijack the cellular machinery to clone themselves, and then multiply until the host cell literally bursts.
Oh my God.
Yeah, the tachyzoites then sweep through the bloodstream, infecting new cells and causing the initial acute infection. And crucially, because they are actively moving and multiplying, they are the form capable of crossing the placental barrier during pregnancy.
Okay, but our immune systems don't just sit back and let the tachyzoites destroy everything, right?
Right.
We fight back, which forces the parasite into its third form, the bradyzoites. Brady meaning slow. I like to picture the immune system acting as this high-security prison guard here.
That's a really good analogy. The cellular mechanism behind it is fascinating. The immune system's T cells constantly patrol the perimeter of the infected tissues.
Right.
They release specific chemical signals like interferon gamma, which put immense stress on the parasite. The tachyzoites sense this hostile environment and realize, hey, we can't win a direct war here.
So what do?
They actually change their gene expression, slow their metabolism to a crawl, and pack themselves tightly inside a protective cyst wall within our brain and muscle tissue.
It's hiding out.
Exactly. And as long as those T cells keep the chemical pressure on, the bradyzoites just stay dormant.
So because it has these 3 highly specialized forms, it has multiple secret back doors into the human body. Which brings us to a massive public misconception we really need to bust right now. You do not get this parasite simply by petting a cat's fur.
No, absolutely not. The transmission routes are very specific. You either ingest the environmental oocysts or you ingest the dormant bradyzoites hidden in another intermediate host.
Right. So for oocysts, we're talking about things like changing a cat's litter box without washing your hands.
Yeah.
Or, gardening in soil where neighborhood cats have buried their waste, and then eating a carrot from that garden without scrubbing it.
Exactly. But the bradyzoite route is actually a massive driver of infection, too. That comes from eating undercooked meat.
Oh.
If you eat a rare lamb chop or undercooked pork or venison, and that animal was carrying dormant cysts in its muscle tissue, those cysts can survive your stomach acid, and the infection cycle starts all over again in you.
That is so gross, but it makes so much sense. And the resilience of the oocysts, in particular, poses some really unique public health challenges, especially regarding water systems. The sources highlight this landmark outbreak in Victoria, British Columbia back in 1995.
Oh yeah, that was a huge case.
Roughly 100 people in the greater Victoria area contracted acute toxoplasmosis at the exact same time, and investigators eventually traced the cluster back to the municipal water reservoir.
Right, 100 people getting sick from unfiltered drinking water, and the biology of the oocyst explains exactly how it happened. Heavy rains likely washed feces from infected cougars or feral cats right into the watershed.
And the treatment plant didn't catch it.
Well, the municipal water was reliant on standard chlorination, and chlorination simply cannot penetrate that cross-linked protein wall of the oocyst. So the parasites easily survive the treatment process. You really need heavy physical filtration or intense UV light to stop them.
Wow. So that outbreak proved at a population level that contaminated water could drive a massive sudden spike in infections.
Exactly.
But whether you get it from water, soil, or undercooked meat, we know the immune system usually forces the parasite into dormancy. We call it latent, but as you said, latent doesn't mean gone. It just means contained by those T-cell prison guards. And if those guards are eliminated, the results are devastating.
Devastating, yes. We saw this play out tragically during the early years of the AIDS epidemic.
Right.
Before the development of effective antiretroviral therapies, HIV would systematically target and destroy a patient's CD4 T cells. Once those cells were wiped out, the chemical pressure keeping the toxoplasma in check completely vanished.
Oh, man, so the parasite just wakes up.
Essentially, yeah. Without that interferon gamma suppressing them, the slow-dividing bradyzoites in the brain sensed the drop in immunity and morphed back into aggressive tachyzoites. This led to toxoplasmic encephalitis.
Which is just awful.
It is. The rapidly dividing parasites began actively destroying brain tissue, causing necrotic lesions, severe seizures, and frequently death. It is a really stark medical illustration of how finely balanced our coexistence with this parasite truly is.
So profound immune suppression is one major vulnerability. But the other time an acute infection turns devastating is at the very beginning of life, before an immune system has fully developed. This brings us to the primary medical concern for Toxoplasma today: newly acquired infections during pregnancy.
Right. Clinicians categorize this under the TORCH acronym for congenital infections, which stands for toxoplasmosis, other, rubella, cytomegalovirus, and herpes.
Okay.
The crucial factor here is that the pregnant person must acquire a new infection. If they already have a latent infection from years ago, their circulating antibodies generally prevent the tachyzoites from ever reaching the placenta.
But a primary infection.
A primary infection during pregnancy means the fast-moving tachyzoites are sweeping through the bloodstream, completely unchecked by antibodies, and they can actively pry their way right through the placental barrier.
And the sources detailed this brutal biological trade-off depending on which trimester the infection occurs. Early in pregnancy, the placenta is thicker and less permeable, so the tachyzoites have a much harder time crossing. But if they do manage to cross in that first trimester, the fetus is in such a fragile early developmental stage that the cellular destruction is just catastrophic.
Yeah. That early destruction leads to what history calls the Sabin triad, named again for Albert Sabin. It's a cluster of 3 severe clinical presentations in the newborn.
What's the first one?
First is chorioretinitis. The tachyzoites actively invade and destroy the delicate cells of the retina, and the massive inflammatory response from the body does even more collateral damage, frequently leading to blindness.
The second is hydrocephalus, right, where fluid buildup deep within the brain. The inflammation caused by the parasite physically blocks the narrow channels where cerebrospinal fluid normally drains, causing the head to literally expand under the pressure.
Yes. And the third is intracranial calcifications. When the tachyzoites destroy clusters of brain cells, the body attempts to isolate that necrotic tissue by depositing calcium over it. When you look at an infant's brain scan, those calcifications are essentially microscopic graveyards, indicating where brain tissue was destroyed by the parasite.
It is absolutely devastating. Now, if the mother gets infected late in the 3rd trimester, the trade-off flips, right? The placenta is much thinner, so the parasite crosses very easily.
Right.
The baby might look completely healthy at birth because the major organs have already formed, but the dormant cysts are hiding in their tissues, and the disease can suddenly emerge years later, usually as severe eye inflammation.
And given these severe biological consequences, it makes complete sense why expectant parents panic. But we really have to look at the actual public health guidelines here, because the fear often outpaces the science.
Totally.
Major organizations, including the CDC, are very clear. You do not need to get rid of the family cat.
Which is such a relief.
Yeah.
So does this mean pregnant people just need to be careful?
Hmm.
Because the logic behind the CDC guidance is rooted directly in the parasite's life cycle. Cats only shed the infectious oocyst for about 1 to 3 weeks during their very first exposure to the parasite, right?
Exactly. For most domestic cats, this happens when they are kittens, first learning to hunt mice outside. An adult indoor cat that has already had it is not an ongoing environmental hazard.
Okay, that makes total sense.
The practical advice is focused entirely on hygiene and breaking the transmission chain. Have someone else change the litter box daily because oocysts actually take a few days outside the cat to become infectious.
Oh, I didn't know that.
Yeah. And if you absolutely must handle the litter yourself, wear gloves and wash your hands immediately. Keep your cat indoors so it can't hunt infected intermediate hosts, and never feed it raw meat.
Right.
And, some nations with different culinary traditions take the meat transmission even more seriously. France, for example, has an intense national prenatal screening program. Because their traditional food culture includes a lot of undercooked meat, they routinely test pregnant women to monitor their immune status and catch new infections immediately.
Wow, that's incredibly proactive.
Mm.
So we know Toxoplasma can cause massive physical destruction in a fetus or an immunocompromised adult. But here is where we venture into the wildest, most fiercely debated section of our sources. We know these cysts sit dormant in the brain, hiding from the immune system. But dormant implies they aren't doing anything. A landmark experiment from the year 2000 suggests they might actually be doing some serious neural rewiring while they wait.
Ah, yes. You are referring to Manuel Berdoy's study at Oxford, which investigated the behavioral manipulation hypothesis. The premise is deeply rooted in evolutionary biology. The parasite needs to get out of the intermediate host, in this case a rat, and back into the feline definitive host to sexually reproduce. So it literally alters the rat's brain chemistry to engineer its own transmission.
The experiment itself is just brilliant. Berdoy and his team placed rats, some infected with Toxoplasma, some uninfected, into an enclosure with different scent zones, including areas heavily marked with cat urine. A normal, uninfected rat smells a cat and immediately freezes or flees. It's just a hardwired evolutionary survival instinct.
But the infected rats displayed a complete inversion of that instinct. They lost their innate fear and were observed actively investigating and spending time in the cat-scented zones.
That is bonkers.
Yeah, Berdoy famously called it a fatal feline attraction. Biologically, the parasite forms cysts in regions of the rat's brain like the amygdala, which processes fear. It appears to alter dopamine production, effectively dampening the fear response and replacing it with curiosity, drastically increasing the likelihood the rat gets eaten.
It is terrifyingly elegant, and because this rodent research is so robust and has been replicated, it didn't take long for the scientific community to look at the massive numbers of human infections and ask the obvious question. If it rewires a rat's brain to change its personality, what is it doing to the 2 billion humans carrying these exact same cysts?
And that leap from rodents to humans ignited a massive academic debate. You have researchers like the evolutionary biologist Jaroslav Fleg, who has spent years publishing data suggesting latent Toxoplasma infection actually alters human behavior.
Really? Like how?
His studies argue that infected humans show slower reaction times, an increased propensity for risk-taking, and statistically higher rates of traffic accidents.
And it doesn't stop at traffic accidents. Here's where it gets really interesting. The psychiatric epidemiologist E. Fuller Torrey published highly publicized meta-analyses showing a statistical correlation between people diagnosed with schizophrenia and positive Toxoplasma antibodies. It essentially revived the controversial idea that severe mental illness might have an infectious trigger.
Yeah, but this is where we have to pause and critically evaluate the methodology.
Right.
When you read a headline claiming a cat parasite causes schizophrenia, the scientific nuance is completely lost. The researchers pushing these theories are identifying statistical associations, but association is absolutely not causation.
That is exactly what I kept thinking as I read these studies. You have to look at the confounding factors. Doesn't testing positive for Toxoplasma just correlate with other lifestyle factors? Like if you live in a lower socioeconomic rural area, you might be more likely to have outdoor cats, eat unwashed produce from a garden, and drink unfiltered well water. That means you get the parasite. But living in that same environment also means you might have less access to psychiatric health care, or you drive on more dangerous, poorly lit roads, which leads to the traffic accidents.
That critical thinking is exactly why the scientific community remains so divided on the human data. When researchers design massive, highly controlled studies to account for all those confounding variables, the dramatic claims of mind control tend to completely evaporate.
Makes sense.
A prime example in our sources is a 2016 study by Karen Sugden and her colleagues. They utilized the Avon longitudinal study of parents and children in the UK, which is this massive data set tracking thousands of individuals from birth.
And they specifically set out to test the viral crazy cat lady narrative, right?
Yep.
The idea that childhood cat ownership or toxoplasma infection leads to psychotic symptoms or severe personality changes later in life.
They did, and because they had such a rich data set, they could statistically strip away the noise. They controlled for family wealth, environmental factors, parental history, and education.
And what did they find?
Once those confounders were accounted for, they found zero meaningful association between toxoplasma infection and schizophrenia, or even increased risk-taking. It directly undercut the smaller, sensationalized studies that had fueled the media frenzy.
It's a massive lesson in media literacy. Journalists love to put the rock-solid science of the rat experiment right next to the human schizophrenia theories as if they are equally proven. The mechanism in rats is an evolutionary fact, but the personality changes in humans remain, at best, a highly debated hypothesis heavily muddied by our complex environment.
Exactly. Extrapolating directly from animal models to human behavior is always scientifically perilous. A rat's brain is designed to respond to predator odors in very specific ways. Humans simply don't operate on those same instinctual olfactory loops.
Synthesizing everything we've explored today on this deep dive, Toxoplasma gondii really forces us to hold two wildly different realities in our heads at the exact same time. On one hand, it is a formidable, highly destructive pathogen. It is a leading cause of death attributed to foodborne illness, a severe biological threat to the delicate cells of developing fetuses, and a major danger to anyone with a compromised immune system.
Yet, simultaneously, for millions and millions of healthy people, it is nothing more than an invisible, silent house guest. It enters the body, gets forced into dormancy by our patrolling T cells, sets up camp in our tissues, and never causes a single symptom for the rest of our lives.
It is wild to think about.
It really is. And if we step back and look at this organism through the evolutionary lens we started with, I want to leave you with a final puzzle to mull over. We've established that humans are a biological dead end for toxoplasma. We have entirely removed ourselves from the food chain. We are no longer preyed upon by large felines.
Right, so the parasite is completely trapped.
So if it is true that these brain cysts are actively trying to manipulate host behavior to draw prey toward a feline predator, are humans inadvertently acting as massive walking storage vaults for a confused ancient protozoan?
Oh, wow.
A parasite quietly sitting in the brains of 2 billion people, altering our dopamine, waiting patiently for a giant predator that is never, ever going to arrive.
That is an incredibly haunting thought to end on. You started listening to this deep dive thinking about a simple medical diagnosis, looking for clear lines and easy answers. Instead, we found an ancient, arc-shaped stowaway that is as medically complex as it is evolutionarily tragic. Thank you for joining us on this exploration of the microscopic world. Keep questioning the headlines you read, keep washing your hands after you garden, and we'll see you next time.